For us, it's really about creating a product that will be highly differentiated from everything else that's out there. And that's really what we've tried to do with Senti 202 with our Logic Gate technology, trying to design it to go after an indication where other drugs really can't fill that gap. And so that's allowed us, fortunately, to be able to recruit patients and recruit clinical sites with strong interest in the program because this is quite a unique product that we've developed in AML. There's nothing else like it.
Hello, and thank you for joining us for the Virtual Investor On Demand Summer Spotlight What's Your Story series. So, my name is Janine Thomas. I am CEO of JTCIR, and today we are featuring Dr. Timothy Liu. He is co founder and chief executive officer of Senti Bio, and he will take us through his story of how and why he is CEO of Senti today. So, before I turn it over to Tim, I'd like to remind you that the company will be making forward looking statements, and I encourage everyone to view the company's website at sentibio.com as well as the SEC's website for their latest filings and information. Okay, Tim, over to you. We want to hear your story. So tell us a little bit about yourself and your journey to CEO.
Well, thanks a lot for the opportunity to be here. I was trained originally as an engineer and scientist. I went to MIT, was an undergrad there in computer science, and eventually made my way over into biology. Ended up doing an MD PhD also at MIT, and at that time, really was focused on figuring out strategies for engineering what we call gene circuits into living cells. I started my lab at MIT as a faculty member in 2010 and spent about 12 years on faculty there. And during that time, we solved a lot of the open issues on how to engineer cells, how to get them to sense their environment, make a computation, and actually do something with that information. And what we were always really passionate about once we had solved some of those basic problems was then to try to figure out what we could do with this technology. And my heart and interest has always been in trying to develop new therapeutics for unmet clinical needs. And it was right around, towards the latter half of the 2010s when we were thinking about, applications of this for treating diseases like cancer. And that's when I took a leave of absence from the university and decided to really jump in full time into Senti and try to take this technology, apply it to treat diseases like acute myeloid leukemia, and then advance these programs to the clinic.
Excellent. Thanks for that, Tim. So, just thinking of going from university to the transition as CEO, what was the biggest transition for you or the biggest change from that specific environment?
I think the biggest thing for us was really now faced with the challenge of actually implementing and creating a drug that was going to move into humans. That certainly posed new challenges and new opportunities for growth for me personally, but also for the technology we were working on. As many of you may know, in the university, we come up with ideas, we can develop the ideas, we can publish the ideas, and really our focus is on trying to develop novelty in the universities. But once we really get down to turning these into an actual drug, that's really when the brass tacks comes in. We have to think about how do we actually create a drug that's going to be unique and fill an important medical need. So there's a lot of discussion with clinicians, other people in the drug development space, really trying to fill a gap that we know will, address a key unmet patient need. There's the manufacturing aspect. How do you actually turn this product, which has a lot of new technology in it? How do we actually make this something that we can actually manufacture and actually safely put into patients? So there's all of that work that needs to come together, and then certainly, aligning, the team as well as investors behind the story to try to push this sort of technology forward.
So when you think about that big leap that you made, Tim, and you think about the potential to help patients, what motivates you every day, and how are there stumbling blocks that are frustrating getting from, kind of thinking about the concept? It has to be because I'm sure you want to help patients, and that's the overall goal, right? So, like, what are the biggest frustrations in that area?
Well, we'd always love to try to go faster and do things more quickly, but for us, it's really about creating a product that will be highly differentiated from everything else that's out there. And that's really what we've tried to do with Senti 202 with our Logic Gate technology, trying to design it to go after an indication where other drugs really can't fill that gap. And so that's allowed us, fortunately, to be able to recruit patients and recruit clinical sites with strong interest in the program because this is quite a unique product that we've developed in AML. There's nothing else like it that's being in the clinic currently. That being said, as we bring these programs forward, there's a ton of work that has to be done in terms of manufacturing the product, It takes time and money and people really coming around the table, figuring out how to make these new therapies, how to release them from the manufacturing with all the quality control that we do around the product, and how to get this into patients. And so that's certainly something that took longer than I had initially expected when we were moving this from the bench to the bedside. But it's something now that we have nailed down. I'm really proud of the team coming together to try to make that a reality.
Tim, what is the biggest lesson that you've learned as CEO of Senti Bio?
Yeah, I think it's a common lesson of anyone who's worked in science, which is that there's a ton of unknowns in biology. We still only have scratched the surface of what we understand about how biology works and how diseases happen. And so, anytime you try to develop a new therapeutic that we want to move forward, I think the thing to always keep in mind that I've learned is not that we're always going to be humbled in some way by the complexities of the human body and how biology has been shaped. So even though we've made tremendous leaps and bounds in terms of our ability to engineer cells and make cells and turn these cells into smarter drugs, there's still a lot more that we could do in terms of improving the performance of the product or sort of improving the engineering process. And that's something that we strive over time to continue to improve in the field.
Well, your journey is certainly impressive, and the work that you're doing is, extremely impressive as well. So, for our audience, what do you think is one thing that they would be surprised to know about you?
Well, this is kind of a little bit of a funny story. I did my PhD with one of the early pioneers of synthetic biology, a man named Dr. Jim Collins. He was quite brave to take me on at the time. When I first got into biology, I knew nothing about how to work in the lab. I had been trained as a computer science person before, but he took me on and actually took a leap of faith. And so I spent hours and hours in his lab, trying to learn the ins and outs of how to manipulate molecular biology. And it's actually something that took quite a long time. I spent so long in the lab sometimes that I think I even forgot about other things that were happening. And so one of the infamous stories during my time as a PhD, which many of my lab mates continue to joke with me about, is one time I was I bought some food I had left in the fridge, and then I decided to try to reheat basically these tortilla chips in the oven. And then I went into the lab to work, and I got so engrossed in what I was doing at the time that actually all of the tortilla chips caught on fire. And so there was a bit of a fire in the kitchen that I had caused, and we had to really rush and get the fire extinguisher and put it out. And it was pretty embarrassing at the time. So, but fortunately, I ended up making it through. Coming out with a good thesis and some scientific work. And actually, Jim, to his credit, continued to stick with me, and we've been close colleagues ever since. He's a board member and co-founder of Senti Bio, so I don't think that left too much of a bad taste in his mouth. But it's something that I don't think many people know that that happened to me when I was going through my scientific training.
Well, you have to have some drama, right, associated with the whole
Yeah,
journey. It's a good story to share.
exactly.
Um, thank you for sharing that. So, before we close, Tim, now as you reflect on your journey to this point, how do you think your story and leadership positions the company to create stakeholder value?
Yeah, so I think at the heart of it, what we are trying to do is make drugs that are completely different than what's out there. If you think about the cancer drugs that we have available today, one of the biggest challenges is that cancer drugs always are fighting between this window of being able to kill the cancer while not having too much of a negative effect on the rest of our body. Chemotherapy, T cell engagers, antibody drug conjugates, and even the existing CAR T cells are really struggling with what we call that narrow therapeutic window. And there's only so many targets that can be used with the current drugs because they require really as much of a clean separation between cancer cells and healthy cells as possible. That leads this whole other universe of cancers that we can't treat well. And what Senti aims to do is really fill that gap. Using our logic gate technology, we can recognize one or more targets on the cancer cell and kill, but very differently from others, we can recognize the healthy cells and protect those healthy cells from being killed. The goal here is to protect the healthy tissues from being affected. And because of that, we believe we can drive stronger anti-cancer activity as a result. So if we're successful in this, we think, in acute myeloid leukemia, there's certainly tremendous opportunities there in the relapse refractory patient population, but also newly diagnosed AML and potentially even into diseases like MDS. But we can also take the same technology and apply it to the vast numbers of solid tumors that are out there that are not adequately treated today. And that's really the mission of what we're doing here at Senti, which is to, number one, showcase that this technology can translate into novel therapeutics and then ultimately deploy this to as many patients as possible.
Thank you so much. Definitely an impressive journey. I loved hearing your story, and thank you for sharing. Certainly an exciting time ahead for Senti Bio, so we'll definitely stay tuned. I'd also like to thank our listeners for tuning in today, and if you're interested in what you heard today, I encourage you to visit the company's website at sentibio.com for more information. And on their website, you can sign up to receive their alerts and follow their social channels to stay current on the latest information. If you'd like to hear more of the Virtual Investor Summer Spotlight What's Your Story webcast, we encourage you to visit our website at virtualinvestorco.com. Thanks everyone for joining and wishing everyone a great rest of your day.